Hi, everyone. Thanks for joining today. We have Dr. Tristan Bepler joining us today. Tristan is a machine learning scientist, group leader of the Simons Machine Learning Center at the New York Structural Biology Center, and CEO and co-founder of OpenProtein.AI. He received his PhD from MIT, where he was advised by Bonnie Berger. Tristan developed the first protein language models for learning protein sequence representations and their application to protein property prediction. He continues to be fascinated by evolution, biophysics, and large-scale machine learning methods. He's also passionate about machine learning methods for understanding protein structures in native states and accelerating cryo-EM. And with that, I'll hand it over to Dr. Butler.
Thanks. Thanks for the introduction, and it's great to be here to present. Let me share my screen, make sure you guys can see this. I hope that looks is working. Okay. Um, so what I wanted to talk about today is some new work we've been doing in protein language models at OpenProtein. And so specifically the next generation of protein language models, designing proteins by learning from evolution, structure, and function. So to give you kind of an overview of what I'll talk about, a little bit of a whirlwind. Hopefully we'll keep things on time, but starting with some background really in proteins and biological sequence modeling, um, why we care about sort of understanding statistical distributions over protein sequences. And then that will segue into a brief intro to protein language models and talking more about some of the earlier models and approaches to the protein language modeling problem and their limitations. And then that will motivate sort of our new protein language model, PoET-2, um, and some of the design decisions we made around this model in terms of its multimodal capabilities and what we wanted this model to be good at and able to do. So we'll talk about some of the use cases in zero-shot variant of prediction and like generative protein design, but then also applications of PoET-2 to learning sequence property relationships, primarily when in the context of or like through the lens of thinking about how we can engineer proteins more efficiently. And then I'll end with some conclusions and forward-looking ideas. So, as I'm sure everyone here is already aware, proteins are the molecular machines of life. They are polymer chains of amino acids. Um, the specific sequence of those amino acids is encoded in the sequence of genes. And so, when we're talking about designing proteins, really what we're able to control is this sort of linear chain amino acid sequence. And that linear chain of amino acids folds into a three-dimensional structure based on the physical interactions between the side chains of the amino acids in this chain, and based on the three-dimensional structure that the protein forms, it carries out some kind of biological function that could be catalyzing enzymatic reactions, binding to things, giving the cell structure. Essentially, proteins are responsible for the majority of function at the molecular level of life. And one thing that makes proteins look really interesting is that because of this sort of nature of being this linear chain of amino acids, there's a combinatorially large sort of design space of possible proteins, right? If we think about how many different possible amino acid sequences there are, even in a relatively short protein with 64 amino acids, there's already more possible sequences than there are atoms in the universe. So this is a really massive potential design space. And most of these sequences, though, don't really do anything. And they probably don't do anything specific, like, right? So if we're thinking about wanting to, say, design an enzyme that carries out a specific reaction, right? There's some vanishingly small number of sequences that might do that. And so the key challenge in protein design really is thinking about, well, how can we identify or how can we actually generate these sequences that will have these specific functions, from this sort of huge, vast space of possible sequences. And um, although this seems impossible because the space is so large, how do you find like that one sequence? Well, it turns out that um, actually, you know, we are able to do this surprisingly well. But mainly methods to do this rely on, say, being able to screen very large numbers of sequences or start from some kind of protein that already does something kind of like what you want, and then be able to make a lot of mutations to that protein to kind of basically, do some kind of like in the lab optimization process to adapt that sequence. Um, and that's really useful, but it's limited. And ultimately, the question here is, what is this space of protein sequences that actually does something useful, right? Like, how can I make a prediction about whether a protein sequence will still be functional, whether it will still be stable, whether it will, still express, right? And ultimately, a lot of the challenge there is thinking about data. Where are we actually going to get sort of the amount of data required to be able to learn those properties? But fortunately, evolution has basically been working on this problem for a very long time. And so we have this vast resource of natural protein sequences that have been selected, over billions of years of evolution for function, for um, carrying out specific functions that confer a fitness advantage to the organism that has this protein. And so we can see sort of the signatures of function and of this selection process in these distribution, like the statistical distribution of sequences that we observe in natural protein sequence databases. So if we look at, say, a multiple sequence alignment of a protein family, right? Like you can immediately see things like, oh, like what positions of this protein are under heavy conservation, what positions are mutable, and that tells us something about the functions of these proteins. And this is not a new idea. These kinds of statistical sequence models have been used for decades to model protein families and kind of understand what are these like functional constraints on or structural constraints on protein sequences. So there's historically, ways that this has been approached is using things like, say, like site-wise factors. So like I was just showing, you can build a multiple sequence alignment and you can just count, how many amino acids occur at each position. And that actually describes a generative model of a protein sequence where, we're sampling basically the amino acid in each position independently from this distribution defined by, our set of homologs. But we can also think about actually modeling more complicated correlations between positions, say like pairwise factors with things like POTS models, to actually explicitly model, the joint distributions over the amino acids at pairs of positions. And then even more complicated like latent factors models or other models that are actually trying to capture higher order distributions. And what was really kind of like fascinating is that actually if you say fit a model like this on the natural homologs of a protein or like a natural protein sequence family, and you actually then take that model and you score mutations in, say, some starting sequence, you feed that wild type sequence through the model, you get its probability, and then you feed all single mutations or something like that through the model, and you get their probabilities, and you compare. You basically like compare like the log likelihood ratio of those, and it turns out that this is highly predictive of actual experimental data. So if we look across large amounts of, say, like deep mutational scanning data sets where people have basically gone to the lab and actually done these experiments where they make every mutation, every single mutation of a protein, and then measure, what the functional outcomes of those are, right? Like whether an organism with that protein still survives or whether that enzyme, what's like is still active, or like what the catalytic rate of that enzyme is, or whether the protein still binds, depending on what protein we're talking about. It turns out that these kinds of basically like family-based generative models, their probabilities are predictive of this lab data, right? And that's because they're picking up on these like functional constraints on the protein sequences that are being learned from the natural proteins in these kinds of evolutionary databases. Okay, so this was a really interesting observation, and this basically is what this kinds of observations underpin a lot of the early work in things like protein homology search, like profile HMMs, protein structure prediction, and basically ideas around using things like multiple sequence alignments or PSSMs as representations of protein sequences in like early machine learning models on proteins. But ultimately, family-based models are limited, especially if we're thinking about directly fitting a model, like a generative model on this set of proteins by available homology information. So if we're thinking about, say, a protein of length L, if you want to learn pairwise couplings, in principle, you need something on the order of like L squared sequences to actually be able to do inference on, all those parameters. And so we're talking about needing, enormous numbers of protein sequences. So it's quite surprising actually that these models work as well as they do, but they are heavily limited by, the requirement of having these very large numbers of homologs. And the other problem with these is we're not really learning like general principles of proteins, right? If you think that, hey, there's some like core biophysical principles here that are determining things like, beta sheets and alpha helices, like those are really like core notions of a protein. If we're training models, like generative models on protein families, it feels like we're leaving something on the table there in terms of our ability to really understand like these other kinds of like more general principles. And of course, if you wanted to use this in a more practical sense, you have to train this then like on a case by case basis. So, this idea kind of really, or this problem motivated some of the early ideas in protein language models, because ultimately, the core problem here was, well, how can we actually develop a generative model of protein sequences that we can actually train on all protein sequences, that we know about, in a way that we could potentially say like distill out some core principles of proteins that we're actually then can access because we can train on like this entire universe of natural protein sequences. And there's hundreds of millions of proteins in UniProt. If you go to metagenomics databases, we're talking like billions of potential proteins out there that we can learn from. And so, there's a few ways we could approach this. So we could think about training, say, like autoregressive models. So where we are modeling the probability of a protein sequence autoregressively. So we factorize it into, say, like the probability of the first amino acid times the probability of the second amino acid given the first amino acid times the probability of the third amino acid given the first two amino acids, et cetera. So we can just factorize the probability of a sequence that way. And if we train a model, say like a recurrent neural network or a transformer specifically to solve that problem, then actually, now we have a model that's very flexible in terms of being able to train on like every protein, potentially unaligned proteins, right? We don't have to care about alignments anymore. So that has a lot of nice properties. But if we actually want to be able to think about using, say, this model to derive some kind of featurizations of the protein sequences, as like a pre-trained foundation model for some kind of downstream learning, then ideally we would really want, say, the embedding of like the ith amino acid to actually capture something about its context in the whole protein sequence and not just can be conditioned on like the previous amino acids. And so, we could build, say, early, a bidirectional language model where we have a model that runs both left and right. But what everyone does today actually is mass language modeling, where essentially you can take a take a sequence, randomly replace some of the amino acids with X, and train the model to look at the entire sequence in that whole context and try to recover sort of what the distribution over what that amino acid is, and therefore like what the distribution is. So we can think about this if you're familiar with like profile HMMs, right? Like autoregressive language modeling, actually profile HMMs are autoregressive language models. But they have a really nice formulation where you can actually like resolve this. You can do what's called posterior decoding, where you basically calculate like the probability of some kind of observation at a position marginalizing over all the other positions. And so that's essentially what mass language models are doing. And these have been very powerful. ESM, classic example here of a large mass language model where these models do learn very interesting properties of proteins. So if we look at the attention maps within ESM, actually, they correlate well with residue-residue contacts in the actual protein structure, right? So the ESM model is actually picking up on these residue-residue correlations as a function of the sequence. And we can kind of pull that directly out of the attention maps in the transformer model. But these protein language models like ESM are really heavily tied to the number of parameters in these models. So we have very large sequence databases. And so you can just make these models larger and larger and larger. You put more parameters into the model, and they're able to capture sort of more and more and more of that statistical distribution. And that does make them better at some problems. But it also makes them increasingly prohibitively expensive to train, but also not just to train, also to use. And so really we run into this like limitation, which is that training a protein language model like this on a database like UniProt is really requiring this model to memorize the entire protein universe. These models don't really generalize in the sense that if you now feed in a protein from some completely unknown protein family and ask the model to be able to like understand, the nuances of what are the statistical constraints on this protein, like what is that fitness landscape, it can't really do it, right? It's learning about all protein families that we know about, distilling all of that down into the parameters of the model directly, and trying to sort of essentially encode all of those fitness landscapes. And so that does confer some degree of generalization, but ultimately it has to memorize the entire protein universe. The other problem with these models is they're not really controllable. If we're thinking about a generative model, like a protein, like a model that you would want to use to actually design a new protein, well, I don't want to design a random protein from UniProt, right? I want to generate a specific protein with some specific properties, right? I want to generate an antibody, or I want to generate, a beta lactamase, right? I want to generate specific proteins for specific purposes. And so we need to be able to guide these protein language models with, structural or functional constraints in some way. And then, of course, we have the scaling problem. And so, that's really what motivated us in terms of thinking about, now getting to PoET-2, in terms of thinking about, how can we overcome some of these limitations in the way your protein language models work. So the key idea here really was that we wanted to build a multimodal protein language model that captures both all of this information in these like sequence databases, but also confers a way to control the model or to like specialize the model directly to specific protein families. And so we were basically thinking about with PoET-2, it's kind of a hybrid between a general purpose model, like these kinds of like a fully unconditional protein language model, and a family-based model. And so when we're thinking about like the evolutionary aspect of this, essentially the idea is, well, what if we train a protein language model to actually be able to solve the family modeling problem? So the key idea there is, if I show my model 3 examples of beta lactamases, it should be able to generate the 4th beta lactamase and the 5th beta lactamase and the 6th beta lactamase, right? So this ultimately is like the kind of like low in protein family modeling problem, which is if I get to see a few homologs, can I generate other proteins from that same distribution? And we can actually then train one model to do this across sort of the entire protein universe to make it general purpose in the sense that it can, then understand these kinds of general protein properties, but also then make it very easily to control because I can directly kind of like show the model protein sequences and it can do in context learning then with no parameter updates. It just basically directly conditions on those sequences to infer what that fitness landscape is and generate new sequences from that distribution. Um, but at the same time, we wanted to also encode sort of additional controllability capabilities into this model. So that was sort of the idea with the original PoET model. And so we also include things like directly being able to condition on structures. So for example, being able to say, okay, what if I also am able to provide from, say, maybe I have some known structures. Can I also include those structures in kind of like this context that I show the model to inform its generative distribution? But also, if we're thinking about using these models actually for things like de novo protein design, I actually really want to be able to then say, I actually want to generate a protein like with this structure. And then maybe I want to condition on, say, some kind of like set of proteins with a function I'm interested in, but then I want to generate a protein with this specific structure or that has these specific sequence constraints, say like active site residues. And so we introduced also this notion of like the query input here, where basically this allows you to specify more like direct, like programmatic constraints on what is the protein sequence that will be generated by the model if you were to sample from it. And then on the other side, we have the problem of thinking about, representations. So if we want to sample proteins from a model, we want it to be generative, then we really care about modeling sort of that joint probability distribution over all of the amino acids in the sequence and being able to sample from it. And so autoregressive models are really the ideal fit for that problem. But if we want to be able to use the model for doing representation learning, we want to be able to bidirectionally encode the whole sequence. And so for that, we really want a bidirectional decoder. So there's this tension of like, well, do I want an autoregressive model or do I want a bidirectional model? And so what we did with PoET-2 was say, why not both? So we basically have both autoregressive and bidirectional decoders. And kind of part of the secret sauce here was that these decoders are actually coupled. So it's actually the same decoder parameters, but you just change the basically like the attention masking to switch it between causal and bidirectional modes. And so, what this actually produced is a model that is able to like learn general principles, but does it actually have to memorize all the protein families in the database? Because it actually gets to see some examples from the protein family in order to then sort of extrapolate, what are the constraints on these proteins from those examples. It's able to model things like higher order mutations and indels because it's fully autoregressive. And it's generative and controllable via prompts. So it basically can do in context learning. So you can show it examples of, say, a human heavy chain antibody sequences, and it becomes a generative model of human heavy chain antibody sequences. You could show it examples of beta lactamases, and then it's a generative model of beta lactamases, right? Without having to change the parameters in any way. But it also then has this like representation learning mode, which ends up being very useful for downstream problems, especially because you can also use in context learning with the representation learning part. So you can basically like focus the model on specific families without changing the parameters, just by changing the context that is used to sort of like embed your sequences of interest. So to kind of dive a little bit deeper on what also this kind of like query and context notion allows the model to do, we can think about like different types of input specifications that we can provide, and essentially then like what problem the model is then solving. So one thing that is, I think, like really exciting about the way this is structured is that it does give a lot of flexibility in those problems. So the model can generate sequences basically unconditionally from a protein family where you only give context with no query, right? So it's just like sampling from that fitness landscape. You can provide sequence constraints. So like I want to sample sequences that have specific residues at specific positions. But then you can also provide structure constraints, right? So you can do like full inverse folding, classic inverse folding. You can do homology conditioned inverse folding. You can do partial inverse folding, say for motif scaffolding, where you provide part of a structure. And you can do a mix where you provide partial structure, partial sequence. And the model can basically generate sequences fulfilling, any of these kinds of input conditions, right? So it gives a lot of flexibility and like composability in terms of what kinds of problems you can solve. Okay, so that's great, but like getting into some of the actual results, right? One thing that I think we were really also thinking about when we were building PoET-2 was, yeah, as I mentioned, how can we make these models more efficient, right? Scaling laws run into the practical barrier of like, I just don't have more GPUs. I can't spend $1 billion to train this model. I can't spend $1 billion to use this model, right? And so one of the things that ended up being really powerful about the way PoET-2 was structured is that this in-context learning notion, removing the need to sort of like memorize the entire database, means that actually PoET-2 can performs like remarkably well with very few parameters. So if we think about basically looking at a generative modeling task here, so we're looking at it's just perplexity of held out UniRef 50 sequences. So this is perplexity is a measure of sequence recovery, so lower is better. PoET-2 is actually able to achieve perplexities that if we were to extrapolate out this kind of like ESM2 a scaling curve would require something like 3 trillion parameters in like a typical mass language model. And this also extends actually to structure. So if you use PoET-2 to predict residue contacts, it actually performs like 10 percentage points better than ESM2 3 billion, despite having only 182 million parameters. But what I think, what I'm really excited about and sort of what I spend most of the time talking about is what this does for our ability to predict the functional consequences of mutations and actually design proteins, both using sort of just direct generative or like zero-shot variant effect prediction kinds of approaches, but then also looking at using PoET-2 as a foundation model for doing supervised learning or like sequence property learning. So first, I'll take a look at zero-shot variant effect prediction. So this is basically the problem of given a protein sequence and optionally a structure, we could always get a predicted structure if we want, predict whether a mutated version of that sequence will be more or less functional. And so we're going to do this based on the likelihood of that sequence compared to the wild type. So this is exactly that same kind of framework I mentioned at the beginning around, if we have a generative model of the protein sequence, we can basically look at those relative fitnesses as predicted by the log probabilities of those sequences in order to predict whether a mutation is likely to be beneficial or harmful to the function of that protein. And one reason to care about zero-shot variant effect prediction, you could ask, okay, great, this is looking at like whether I can predict the functional effect of a single mutation. Does that tell me whether I can actually sample, say, or like design a functional protein with this model? If we're thinking about that, this probably is one of the best benchmarks for that problem, because ultimately what we want to understand is, do sequences that are given high likelihoods by the model, like are those sequences functional? Or are those sequences more functional than sequences that are given low likelihood by the model, right? Because if we're sampling from the model, ultimately, right, we're interested in sampling high likelihood sequences or identifying what are those high likelihood sequences, and we want those to be functional. And so this basically is a direct assessment of that property. So there's a great benchmark for this called Protein Gem. So this is like a comprehensive um, accumulation of deep mutational scanning data sets from the literature. So they have hundreds of deep mutational scanning data sets in this benchmark, and these basically comprise a huge variety of proteins where people have done mostly site saturating mutagenesis, but some actually have higher order mutations in them, insertions and deletions. There's all kinds of interesting mutations in these data sets, and it spans a huge range of proteins and different functional properties. So this gives us a way to get basically a holistic assessment of do um, likelihood scores given by these models correlate with function broadly, or function in the context of any kind of specific protein that we're talking about. And so what I'm showing here is a table of these correlations. So we're looking at the rank correlation coefficient between the model likelihoods and actual functional measurements across different substitution depth, across different MSA depths. So, and here the MSA depth is interesting to look at because we might expect these models to perform well on proteins that have a lot of natural homologs. So this is high MSA depth, right? If you have a lot of natural homologs, we think, okay, we have a lot of data to learn from, learn this generative model from, and so we probably should be able to fairly accurately predict whether a mutation in that protein will work or not from that model. And in part, this is also interesting to look at because sort of the dream of a protein language model is that it should work on proteins with few to no homologs. Because if we've really learned general properties of proteins, I shouldn't need to see homologs, right? I should be able to see that one protein and say, okay, from all of everything I've learned from all these other proteins, I understand what mutations I can make in this protein. And so it's especially interesting to look at to start how well sort of these general purpose protein language models perform on this problem. So if we look at, say, ESM2 or ESM1V or ESM1B, there's a very steep drop off in performance as we go from proteins with large families, high MSA depth, to proteins with low MSA depth, right? Which shows that really, we're not actually, these models are very focused on these large protein families. And they're not actually doing that well at understanding the fitness landscapes of sort of orphan proteins or small protein families. But PoET-2 performs actually very well in that regime, right? It's able to extrapolate from very small numbers of proteins to be able to predict this kind of fitness landscape. And another thing that I think is quite interesting to look at is PoET-2 is actually very good at predicting the functional effects of insertions and deletions. And that's quite unusual because most models aren't autoregressive. They're not trained to be able to do insertions and deletions. If we look at some other breakdowns here of PoET-2, we also noted that PoET-2 actually performs really well at predicting the effects of combinatorial mutations, especially with very large numbers of combinatorial mutations. And this is another benefit actually of the autoregressive formulation, which is that we can actually calculate in closed form sort of that joint probability of all of those mutations being in this sequence. And so we get a better assessment. We don't have to assume any kind of independence between the positions or something like that to then aggregate those probabilities. And PoET-2 also performs remarkably well on, say, also on predicting functional effects of things like viral proteins, which are typically hard for these kinds of models. So what else can we do with this kind of in-context learning? A lot of the highlight here is that this is really about conferring sort of a way to very creatively control what the model will do. So the model's trained to be able to understand homologs, right? Extrapolate from homologs, but you don't have to feed homologs into the model as this kind of context, right? We could condition on the naive human antibody repertoire sequences, for example. And if we do that, we basically then end up with like a general purpose human antibody generator. And it turns out that likelihoods of that model actually correlate really well with things like um, immunogenicity um, of antibodies, right? So this is an analysis of anti-drug antibodies where basically we have antibodies that have gone through clinical trials, and we know what percentage of patients actually developed like an anti-drug antibody response. And if we look at the score that these antibody drugs would receive, under this like human antibody distribution, it actually correlates very well with percent ADA, right? So more human-like, higher likelihood antibodies are less immunogenic. But this also correlates well with things like developability properties. So if we look at an analysis of the antibody likelihoods for now, we're looking at antibody mutations versus change in thermostability. Antibodies with higher likelihood actually end up being mutations that increase likelihood tend to also increase thermostability. So what else can we do with this? We can do things like motif scaffolding. So we can. Sorry, I think someone is unmuted. Oh, okay. So for example, we could motif scaffold a truncated nanoluciferase, right? So this is just showing an example, right? Like nanoluc is quite interesting, is a quite interesting protein. It's actually an orphan protein. It has no homologs in any of the natural protein databases. And it's this weird beta barrel protein that catalyzes like luciferase reaction. But if you actually look at what are homologs of this protein, they're mostly fatty acid transporters. So it's quite unusual. It has also this weird extracellular domain. So we might want to do something like, let's chop that off. Let's constrain the active site because we know, these active site residues actually confer the function, and like, let's rescaffold this protein, right? Like rebuild the rest of the protein, right? So we can do something like that with PoET-2. We can also do things that have no like structural relationship, right? So a lot of the focus in de novo protein design has been on things like inverse folding, where we're saying like, okay, I'm going to define the function of this protein through defining a structure that I think will give this function. But for a lot of functions, we may not know what that structure is. For a lot of functions, there may not really be a specific structure that we want this protein to form and fold into. So for example, cell penetrating peptides or blood-brain barrier crossing peptides. What structure does that peptide need to form to cross a membrane? I don't know. There are many different mechanisms by which these peptides could cross the blood-brain barrier. But we can still learn about what those functional constraints are sort of directly by conditioning on those peptides. So what we did here is we actually just took a cell penetrating peptide database, fed that into PoET as our context, generated new peptides from that distribution, tested those peptides for toxicity and blood-brain barrier crossing in zebrafish embryo. And we actually found that we were able to generate a set of non-toxic peptides that actually had blood-brain barrier penetration. No kind of like structural hypothesis here at all. So this kind of in-context learning, and query definition capability really gives a lot of flexibility and room for creativity in controlling what the model does. Okay. Moving beyond sort of this kind of generative modeling and evolution, right? Evolution is a very powerful source of information, but all of these purely evolution-based, purely likelihood-based models have a problem, which is that they model fitness. They don't predict specific properties about a protein. the properties that it's predicting are emergent, if you will, from the selection process that led to the set of sequences that we observe in nature or that were provided to the models or in the context. It defines a fitness landscape. And so that correlates with things like activity and stability and expressibility and pathogenicity, et cetera, et cetera, et cetera. But it is very much dependent on what are those actual selection pressures. And the model, as a consequence, can't predict these specific properties. It's not directly predicting a stability, right? It's not directly predicting a catalytic rate. It's not directly predicting a KD. So to predict those kinds of specific properties, we need to learn those sequence to function relationships in some way. So we need some data that lets us actually basically align our protein representations with those properties, or lets us discover what are the, what is that underlying sort of subspace of our features as learned by this model that encode that property. Okay, so this is where we get into really the representation learning aspect of the model. So this is why having the bidirectional kind of decoder is really important. So here what we're looking at is actually trying to learn from small amounts of mutagenesis data to predict specific properties and use those models to then design optimized sequences. So this is essentially the core problem in protein optimization, which is I want to optimize this enzyme to, say, improve its catalytic rate. I'm going to make some mutations to this enzyme. I'm going to measure what are actually then the catalytic rates of all of those mutants. Can I learn from that to then actually map out that sequence to function relationship and then identify what mutations will maximize my catalytic rate, for example? And so the importance here really is about being able to learn very efficiently from small amounts of data, right? We want this model to generalize as quickly as possible. So the framework for this experiment is exactly that. So we have a training set, which is generally single mutants of a protein with some kind of experimental score. Our training set size, we're going to look at varying from 10 to thousands of proteins. We're then going to featurize all of those sequences with PoET-2, and then train a specialized model on top of that to actually predict our experimental scores. And then we're going to look at using that model to predict those property measurements of unseen variants, and we'll just look at those correlations. And we're going to look at this across all of the data sets in Protein Gym. So this is like an aggregate analysis on hundreds of data sets. One other thing that's important to look at is thinking about what is our cross-validation approach here. There's a few different settings that we might care about. So the first is a random split. So in this case, what we're saying is, okay, I have every single mutation of a protein. I get to see some random subset of those for training, and I'm going to evaluate on the rest. And so what that means is I might see a mutation at a position. Maybe I see a alanine mutation, or I see a arginine mutation at a position, but I don't see an alanine mutation there. And so I'm saying, so I get to see the arginine mutation, and I get to learn from that, but then I'm making a prediction for an amino acid that I haven't seen. And then we have increasingly difficult sort of ways of stratifying the data. So the next is the modulo split. So the modulo split says, okay, I get to see mutations at some positions, but then I have mutations or positions that I'm going to evaluate on where I don't get to see any mutations at those positions, but they could occur right next to a position where I saw some mutations. And then the most difficult split here that requires sort of the greatest degree of extrapolation is the contiguous split, where there's some whole region of the protein where I don't see any mutations. And essentially we're asking, okay, if I don't see any mutations in this whole region of the protein, how well can I extrapolate from mutations in other parts of the protein to sort of understand what then the functional consequences of mutations in this region would be? Okay, so if we compare across different training set sizes, different featurization models, different ways of featurizing the protein sequence in these three different schemes, we get an idea of how effective these underlying protein representations are. Because the idea here is that a better representation is better aligned with these functions, and therefore, if we look across different numbers of sequences in our training set, so that is, I get to see only 10 mutations. How well can I predict, what will happen if I make another mutation? Versus I get to see 50 mutations, I get to see 100 mutations, up to 2,600-ish here, right? Then a better featurization will improve our efficiency across this whole curve. But really, right, what we really care about if we're thinking about in protein engineering is, well, how few mutations do I need to make to be able to kind of like get to my outcome, like have a reasonable predictor. So it's quite exciting, I think, to see how effective PoET-2 is, as a representation learning model, especially for this kind of problem. So we are actually able to improve predictions so much that we can achieve prediction accuracies that would require like 20 times more data if we were using like ESM as our foundation model. And so this kind of in-context learning and the ability to like directly incorporate the evolutionary context like really dramatically improves the quality of these embeddings. So what does this mean for actually doing optimization in practice? Well, we actually can learn from very small data sets to identify promising mutations. So one example is we actually took 30 measurements so PETase is a plastic degrading enzyme. People have been very interested in this enzyme recently because of applications to basically being able to recycle PET. So what this enzyme does is it breaks down the PET polymer into monomeric units. And we scraped together a data set of just 30 measurements. So we have 30 variants of this wild type PETase enzyme. So actually, there have been some natural enzymes that have been found that are able to degrade PET that have basically like naturally evolved in bacteria that live on like trash heaps. So in garbage dumps or actually in the Pacific Ocean, like directly on plastic. And so we have this wild type enzyme that has quite low activity. So people are very interested in optimizing that enzyme specifically to see if we can like actually get this enzyme to have high enough activity to be useful in the industrial recycling process. And so we have these 30 measurements, so measurements for 30 variants scraped from the literature. We trained an activity predictor in exactly the same way I just described on these 30 data points. We use that to then do an in silico screen of a large number of combinatorial mutations of this protein and selected a few to test in the lab. And actually, in parallel, we did this with a collaborator who was doing some rational designs of this protein as well. And so what we found was that we were actually able to find just, in our first round of just 3 mutations, a mutation that increased the activity of this protein by like 4x, and actually was even more active than what at the time was the best known variant. And then we did a follow-up library where we did 10 more variants that all were either equivalent or even higher activity. So this kind of approach actually is very effective for this kind of even learning for this kind of very low in kind of mutagenesis data to drive protein optimization. But we can also use PoET-2 as a foundation model for other kinds of property learning. So for example, we can improve our ability to predict signal peptides and protein secretion, on like a general purpose data set for this problem. We're looking at the signal P6 training dense data set and benchmark. And actually, PoET-2 is able to provide a remarkable increase in accuracy for some signal peptide types. Okay. So I'll wrap up there and hopefully leave some time for questions. So to conclude, protein language models are powerful models for distilling design principles of proteins from evolutionary data. PoET-2 itself is a high accuracy, fast generative protein language model that's also controllable via this flexible constraints grammar. Um, Not only is it generative, we can also use it actually as a foundation model. It captures structural and functional properties of protein sequences in this embedding space that it's learned. And so we can fine tune it or train models directly on top of it to do things like map sequence function relationships for protein optimization, but also do things like general purpose downstream ML. Okay, so with that, I have to thank a lot of people who contributed to this work. I want to especially thank Tim, who worked on the PoET-2 model, and some of our collaborators. Okay, happy to take any questions if there's some.
All right, thank you so much for the amazing talk. We'll take some questions from the audience. I think there was a question from Audit from the chat. Do you wanna ask the question, Audit?
Sure, yeah. Great talk. It was very insightful in the context of just like making these kind of language models and generative models practical in different workflows. One question I had was, and it was mentioned a little earlier, but predicted functionality was one of the metrics that was assessed with regards to the predicted sequences. Is that functionality usually in the form of like how it impacts just downstream kind of cellular processes, or like what was the method for predicting functionality and
Yeah,
assessment?
so Protein Gym has a big diversity of different functionalities in that sense. So some are, yeah, very um kind of ill-defined, I guess. Like it's just a fitness measurement. Like does a cell with this mutant protein survive? Um, or like how frequently do you see it in the population essentially? So it's like essentially a fitness measurement. But then others are more specific. So you have enzyme activities, binding measurements, stability. There's quite a lot of stability data in Protein Gym, expression of the protein. So it spans the whole range.
Okay, yeah, that makes sense considering it's like more specific kind of contexts the sequence can operate within, and then larger ones that are general across different types of predicted sequences. So thanks for
Yeah.
answering that.
Yeah.
Opera, wanna ask a question?
Hi, thank you so much for your talk. I was wondering how much, so you talked about how a lot of these models require kind of larger and larger parameter sets, and you've kind of managed to scale back on that. But I'm also wondering, in terms of learning sort of generalizable patterns about proteins and protein dynamics, whether to what extent you think that we could get information by adding, like more explicitly adding physical and biophysical knowledge, for example, through simulations or through like kind of close, like I'm thinking of like molecular dynamics simulations or something
Yeah.
like that. That, yeah.
Yeah. I think that's actually a very interesting direction. Um, I think the key question there is what is the right way to incorporate that information? I mean,
Mhm.
I think one approach that is really interesting is to think about are there ways to basically change the like neural network architectures of these models to explicitly cap model, something about the physical structure of the protein, or to explicitly incorporate like a force field or something. Although you then have to think about how that relates to your actual like training objective of the model. I mean, the other approach is more like just like, okay, data driven, let's just also train these models to predict structure. Well, why stop at PDB structures? Why don't we train these models to predict structural like thermodynamic ensembles that we generate with molecular dynamics, right? I mean, that's what people are working on, like BioEmu, and we actually have some work on this as well. new work too, looking at trying to predict thermodynamic ensembles. But then really like, can we bring that all the way back into the foundation model and like use that to sort of inform the underlying like representations of the protein that we learn? I think is a very I think could be a promising direction. Yep. I mean, not fully tested yet. So definitely exciting.
Thank you.
Yeah.
I have a question about the model. I think one of the interesting figure was the how you show the model performance with the MSA depth. And
Yeah.
models like ESM2 and ESMC are supposed to be unbiased because they're not training specifically with MSA like art structure. And yet they show like clear correlation as you go. For low MSA depth, it does way worse. I'm
Yeah.
wondering, and I think we saw a similar thing for ESM fold and like AlphaFold as well.
Yeah. Definitely.
I was wondering if you have tried the PoET-2 architecture for the actual structure prediction, and is there any indication that MSA depth also affects the model performance there?
Um, yeah. So good question. We we have been working a little bit on actually trying to build like a full structure predictor on top of PoET-2, although it's still kind of early days in that. So I can't say exactly what kind of like the MSA depth scaling versus structure prediction accuracy of that model is right now. I mean, it it definitely looks promising in the sense that even though PoET-2 doesn't condition on an alignment, it's just unaligned sequences. We can get pretty good structure predictions out of it. I think it needs to, the whole architecture does these days needs a lot of tuning though, I think to get to like AlphaFold 3 kind of level of performance, right? So we
Right.
haven't pushed it all the way yet. But now it'll be interesting to look at that. Yeah, I don't know the answer yet to that. What I can tell you, um, and this isn't about structure. This is going to be about more like functional predictions, like on Protein Gym. So one thing that's really counterintuitive and surprised us a lot was that just increasing the number of homologs you condition on does not improve the performance of the function prediction in Protein Gym. And it does with models that don't use the data as efficiently. But with a model like PoET-2, we actually hit diminishing returns very quickly. And actually, sometimes even hit an inflection point where as you add more sequences, performance starts to degrade. And we don't really see that with structure though. But with the like fitness predictions, we do it. There have been some other works that have observed the same thing. And it probably has to do with something about how uh, if you model like a homolog or like a protein and you look just like too much only at the homologs with a few mutations, that like it's very kind of like myopic then towards that like wild type protein and doesn't understand sort of like the broader distribution of the function. But um, that was kind of surprising. So, but structure doesn't seem to follow that pattern, which is interesting.
I see. Thank you. We're at 2, so we're gonna wrap this up. But thank you so much for your seminar today. It was very interesting.
Yeah,
And
my
thanks
pleasure.
everyone for coming.
Yeah, my pleasure.